Namaste to all students.
Khushbu, myself JV'n Dr. Khushbu Verma, Faculty of Agriculture and Veterinary Science. Today we will discuss about gene transfer techniques, which is microinjection, and this session is related to recombinant DNA technology course of B.Tech FBT and B.Sc BT 6th trimester. It is defined simply as a technique to efficiently and stably introduce foreign genes into the genome of target cells. These methods are also classified into two groups. First is direct method and second is indirect method, or you can say agrobacterium mediated gene transfer method. Direct gene transfer relies on the delivery of large amounts of DNA, while the plant cell is transiently permeabilized by particle bombardment, electroporation, ultrasound, etc. It is mostly used for gene transfer in cereal crops like monocotyledonous and dicotyledons. Agrobacterium mediated gene transfer are based on utilizing Agrobacterium, a pathogen of dicotyledons plants that transfer genes into the plant genome. This is a flow diagram where we can see that direct method can be divided into two types: physical direct method and chemical direct method. Physical method involves microinjection, pressure, bioelastic, gene gun or particle bombardment method, electroporation method, silica carbon fiber laser mediated method. In chemical direct method, it's a PEG mediated method, calcium phosphate method, artificial lipid, protein, dendrimers mediated direct method. And second is indirect method, which is biological method with the help of agrobacterium. Now come to the microinjection, on which we will concentrate in this session. DNA microinjection was first proposed by Dr. Marshall A. Barber in the early of 19th century. This method is widely used for gene transfection in mammals. It involves delivery of foreign DNA into a living cell. Example, a cell, egg, oocyte, embryos of animals. Through a fine glass micropipette, the introduced DNA may lead to over or under expression of certain genes. It is used to identify the characteristic function of dominant genes. The delivery of foreign DNA is done under a powerful microscope using a glass micropipette tip of 0.5 millimeter in diameter. Cells to be microinjected are placed in a container. Holding pipette is placed in the field of view of the microscope that sucks and holds a target cell at the tip. The tip of micropipette is injected through the membrane of the cell to deliver the content of the needle into cytoplasm, and then the empty needles is taken out. This is a figure of delivery of DNA into a cell through microinjection. Here we can see micropipette containing foreign DNA. This is cell, and this cell is holding by the holding pipette, and micropipette is injecting foreign DNA indirect into the nucleus. What are the advantages of microinjection? So no requirement of a marker gene, introduction of a target gene directly into a single cell, easy identification of transformed cells upon injection of dye along with the DNA, no requirement of selection of transformed cells using antibiotic resistance or herbicide resistance marker. It can be used for creating transgenic organisms, particularly mammals. Disadvantage for microinjection is the procedure is time consuming and only a small number of cells can be treated. This technique provides directly nuclear delivery of DNA, avoiding the endogenous pathway, and also ensures that the DNA is delivered intact. Microinjection is suitable for the introduction of large vectors such as yak into the pronuclei of fertilized mouse eggs. DNA delivered in this manner must be very pure, so it needs a lot of preparation as it is necessary to avoid fragmentation. Sharing can also occur in the delivery needle, and large DNA fragments are often protected by suspension in a high salt buffer and or mixing the polyamines and other protective agents. Now transfection of cultured cells is automated with computer controlled micro manipulation and micro injection processes, as well as the automated production of injection capillaries and the standardization of cell preparation procedure. So this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University. Hope you are satisfied with my digital session. If you have any query, then please mention in the comment box. I will resolve it. Thank you.